Welcome, everyone. Thank you for joining us for today's track on life sciences. It's my pleasure to introduce to you Dr. Dino Di Carlo, who is a professor of bioengineering and serves as the vice chair of the department and as the director of the Cancer Nanotechnology Program in the Jonsson Comprehensive Cancer Center at the University of California, Los Angeles. Dr. Di Carlo's research pioneered the use of inertial fluid dynamic effects for the control, separation, and analysis of cells in microfluidic devices. His recent work extends into numerous other fields of biomedicine and biotechnology, including directed evolution of cells, cell analysis for rapid diagnostics, mechanomedicine, next-generation biomaterials, and phenotypic drug screening. He has also been a leader in technology entrepreneurship, having co-founded 5 companies that are commercializing UCLA intellectual property developed in his lab. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Di Carlo's keynote presentation is titled Quantum Diagnostics, From Single Cells to Single Molecules. My name is Judy O'Rourke, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Dino Di Carlo. I will now turn the presentation over to him.
Thank you for the introduction, and it's my pleasure to share the work we're doing on quantum diagnostics. And so the reason we are saying that we're using the word quantum diagnostics is that we're looking at the ultimate levels of information content in biology. And so that's at the level of individual cells and individual molecules, like individual nucleic acids and individual proteins. And so now we have the capabilities to measure at this ultimate limit, and a lot of that has been enabled by microfluidic technology. And so what I'm going to talk about today is what I think is the next step and moving beyond microfluidic technology to make these diagnostics at the ultimate limits of biology more available and adopted across clinical diagnostics and research in life sciences. So, I've worked for a long time in the microfluidics space, and we've done a number of different types of work in this area. And here's one example of a work that we've done on analyzing and developing technology to isolate cancer cells from blood using a microfluidic chip. And so, when you look at this type of chip in the lab setting, you see that these are small microfluidic devices. This is a particular device we call a Vortex trapping device. But what happens is, as these types of devices translate, and although they may have very unique capabilities and provide benefit, they start to look more like this. And so this is the VTX1 liquid biopsy system that makes use of this Vortex trapping technology based off of microfluidics. But now you have all these control systems. You have pumps, you have electronics, you have a number of other elements that make a larger scale instrument required in order to control that really simple microfluidic device. And so even though this has unique advantages in order to isolate, for example, circulating tumor cells in this application, the ability for this to spread across multiple labs into clinical diagnostics becomes more limited because of the need to uptake to buy new instruments, to buy new capital equipment. And so this is a general challenge with microfluidics, is that although we have ability to make small scale devices, which we think should be low cost and easily adopted by many, what it turns out to be is that these microfluidic devices then have to be operated by much larger scale systems. And so that's a challenge that we're working to address with new types of technology. And so I want to talk about how our technology particularly makes use of one of the key advantages of microfluidics, which is the power of compartments. And so the idea here is that in microfluidics, one thing that has maybe driven some of the killer applications in microfluidics, whether it's single molecule analysis or single cell analysis, is the ability to compartmentalize a larger fluid volume and break that up into smaller and smaller fluid volumes. And as you do that, you start to see, so on your left, you can see a kind of schematic where we're showing target and non-target molecules that kind of look like nucleic acids in this case. And as you start to break up that volume into smaller compartments, nanoliter or sub-nanoliter volume compartments, what you start to see is some unique advantages. So for an example, if you look at the volume in the upper right quadrant, you've now completely purified that particular volume from the non-target nucleic acid, and you have also concentrated the target nucleic acid compared to the background volume. The other key aspect is now these small volumes, let's say that you need to create an amplified signal in order to detect these single molecules. That amplified signal accumulates to very high concentrations in that small volume and enables you to detect it very easily. That also is very valuable, let's say if you have a cell that's secreting some signal, and that, or you're lysing a cell and you want to extract material from that cell, that material or that secreted signal becomes at a much higher concentration in that small volume. So this has really driven a lot of innovation in microfluidics, and as I said, some of the killer applications. And here's an example of one application that Bio-Rad has developed, has commercialized, that's called Droplet digital PCR. And so there are kind of a few key steps here. The first step is using microfluidics to create uniform volumes to compartmentalize this larger volume. And again, the uniformity of that volume is also important because you want the reactions that proceed in those volumes to occur at the same rates. And so let's say products that accumulate in those volumes to be at similar concentrations. You then can run a reaction in that volume. And so in the Bio-Rad and Droplet digital PCR approach, you're incubating in order to amplify a nucleic acid using PCR. And then you read out those compartments using some type of fluorescence measurement based on the amplified signal that is accumulated in the compartment. So this looks very simple, and so there's microfluidics and some readout, but what it turns out is actually in order to achieve this, you need new instrumentation, as I mentioned before, right? So you need one instrument in order to generate these droplets, these monodispersed compartments, and you need another instrument. So you need also a thermocycler in order to run PCR, and then you need a third instrument in order to read out those droplets. And so these are all unique instruments that you need to then acquire in order to bring this really powerful digital or single molecule or quantum diagnostic assays to clinical use. And so that's a barrier because of cost, of course, to acquire new instruments. But even in order to try out new things, there's a large barrier that any new clinical lab or a researcher who's trying to develop new assays has to overcome to do this. And this, of course, is for one particular assay, which is a digital nucleic acid amplification assay. But now if you imagine if you want to look at single protein molecules using digital ELISA or single cells, for each of these assays, you now need new equipment. So that's a big issue. So the dependence on new infrastructure and disposables for each of these quantum assays is a challenge for adoption and for this really technology that's out the ultimate limits of what we can analyze in a biological sense to reach humanity in a larger sense, this has been a barrier. So the other aspect of these compartment systems that is important and has been a challenge is you also want, in some of these assays, a solid phase for reaction. So I'm showing here a digital ELISA from the Quanteric Simoa system, where you have a bead that is that has your capture antibody to perform your single molecule ELISA. You also have in single cell RNA-seq, this is an example from the 10x genomics system, where you have a cell in a compartment, but also a bead in that compartment that will capture the mRNA from that lysed cell in order to then perform barcoding of that mRNA and single cell RNA sequencing. And finally, there's a number of assays that make use of a solid phase to capture the secretions of individual cells. And this is showing one example from Chris Love's group, where they're capturing cytokine secretions from leukocytes on a surface that's coated with antibodies. So the solid phase is also an important aspect of these quantum assays. Okay, so the question that then arises is, if we have this barrier for infrastructure with current microfluidic technologies, how can we overcome that barrier? And so can we create uniform small volumes? Uniform is key here as well. And each of those volumes having a solid phase that allows us to create these compartments that have unique advantages, but then also analyze the output of those of reactions that occur in those compartments without new equipment that may be challenging, the new infrastructure that has to be acquired. And so, first one question that I'll ask and answer is, what is the highly sensitive and what is a highly sensitive and high throughput analyzer of fluorescence, which is many of the signals that we expect to see from these digital assays will be fluorescent. That's almost in any research or clinical lab. And one answer to this is a flow cytometer. Right? So these are ubiquitous analyzers, extremely high sensitivity, using photomultiplier tubes to look at fluorescence of single cells. They can measure multiple different colors. And so they have a lot of power, and their presence is ubiquitous in research labs, but also in clinical labs. So can we leverage that as our analysis platform for assays that are occurring in these individual particles or individual compartments? Okay, so the challenge then is not for the reader, but it is how do we create these uniform compartments in a very simple process? So one way to create these compartments is with emulsions. Of course, there's microwells and other things that are used in microfluidics, but emulsions has been really a driver of many of the applications. However, if you just shake an emulsion, shake up oil and water to create an emulsion, that emulsion is not stable. It's metastable. You get non-uniform drops of many different sizes, and those drops will want to coalesce over time. So can we make an emulsion that you just shake up and it creates uniform sized drops, and then each of those drops also has that solid phase that we talked about? And so that is the challenge, and I'm going to answer whether we can do this to not hold you in suspense, and the answer is yes, that we can do this, and I'm going to tell you about the technology that we've been developing, this particle drop technology, in order to make this happen. And so it's useful to kind of step back a bit and think about kind of the fundamentals of these types of emulsion systems. And one way that I like to think about these systems is through something I call a volume energy curve, or a VE curve. If you look at the volume energy curve for a drop of, let's say, water and oil, it will follow this relationship of the energy, the interfacial energy, scaling with the volumes to the 2/3 power. And that's because the interfacial energy scales with the surface area, and the volume is scaling with the radius to the 3rd power. And so what you see is this curve where it's the convexity of the curve is down, and so you have a convex curve for this volume energy curve. And what that convex curve means is that those droplets will always want to coalesce. So two smaller volume droplets will want to merge to create a larger volume droplet, because that will be the minimum in energy. And so you can kind of look through this. If you have a convex curve in the volume energy landscape, and you have two droplets that interact, what you'll see is that those droplets will start to merge to create a bigger droplet and another smaller droplet down to no droplet, so that you accumulate into one larger volume droplet. So that happens with convexity, a convex curve in this volume energy landscape. Now, if you start to add concavity to this curve, that you start to see some really interesting effects. So now two different volume droplets that interact, what they want to do is actually go and partition evenly to two evenly sized droplets that are half of the total volume. And so I've drawn this volume energy curve as having minima in the energy landscape, but it's not necessary. It just has to have convex region and a concave region. And so what you can imagine now is that you start to have droplets that will accumulate at this particular size. So as if the volume of the droplet is too small, they'll start to get bigger and bigger and bigger and merge. And then at some point, as they reach a larger size, then they will start to split in half. And then you can have another region that's convex again, where you would then start to accumulate droplets at this minima, and then one big large droplet. Okay, so this is the strategy that we can start to take to in order to think about how to create monodispersed droplets in an energetically favorable way. Of course, the question then arises, how do we create, in a physical sense, this type of volume energy curve? And so one way to do this is have the droplets interact with surfaces. So if you have a droplet interact with a single surface, it will form a spherical cap on that surface. And that spherical cap will have a certain energy landscape that actually looks convex, similar to a sphere. That convexity starts to change, as you see here, when you have two surfaces interacting and you form a catenoid as the drop interacts between those two surfaces. And so the strategy here is adding more interactions with these wetting microstructures around the droplet in order to change the convexity. So you can imagine as you add more structure beyond two parallel planes of surfaces, then you start to bring this curve down further and further. And as it starts to move down further and further, in order to then transition out to a droplet that's completely full, it will have to, the convexity will have to change again. And so you can start to see this. Just to summarize kind of how this will work is that we need some type of structured particles to modulate this volume energy curve. So here are two volume energy curves for a free droplet that follows this volume to the 2/3 power, and you see a surfactant coated droplet. It still follows this volume to the 2/3 power. The surfactant will just lower the interfacial energy, but still the droplets that are coated with surfactants will still want to coalesce over time. However, if we start to now have a structured particle that has these two parallel plates, but also a edge around it, what you can start to see is you modulate that volume energy curve to have a convex region at the beginning between 1 and 3, and then a concave region, and then another convex region as you go from 4, 5 to 6. And so what I'm showing on the bottom here are models and simulations of how a droplet would fill this type of particle, and then the energy, the interfacial, the total system energy that corresponds to these filling states. And this is if you impose that this is the amount of volume that has to go into the droplet. So what you can start to see is that this type of volume energy curve if you then analyze how this type of, how a volume would split between two of these types of particles, what you see is that at lower volumes, we're in that convex region, and the whole volume will stay either in one or the other of the particle. So that's that first region where you don't get splitting of the droplet, and so you get filling of one of the particles. And then as the volume increases further, let's say this normalized volume of 2, then you can see that the energy minima is actually when the volume splits evenly between the two particles. So that means that now these particles are full, and they will split and fill the next particle. And then if you go to even higher volumes, then you start to see that one particle will fill completely, and the other particle will take the rest of the volume. And so that's kind of goes back to a convex region. But that's really the mechanism on how you can fill particles to this, this uniform controlled volume in an energetically favorable way, so that you can do this with simple mixing processes, ideally. Okay, so we demonstrated some of this using a model system, and the videos here are not going to show, but that's fine. I'll show you some data that comes from these videos. And what you see here is that this is a PEG polyethylene glycol internal layer and polypropylene glycol external layer. So you have an internal polymer layer that's hydrophilic, and an outside layer of this particle that's a bit more hydrophobic. And this is the type of particle that we're using to generate these types of particle drops. And so if you do experiments where you start to split the volumes between macro scale particles, so you can see the volumes here. These are particles that we made using some 3D printing type of molds. You can see that at low volumes below around 600 microliters, that the two particles will split evenly, and you'll get an even volume to each particle. But above about 600 microliters, one particle will kind of maximize, we'll get to this maximum volume and stay there, while the other particle will take the rest of the volume. So you can imagine over large numbers of particles that this leads to a number of these particles all holding the same volume, and then a larger volume around that's held by one particle. So that's on the micro scale. So you can see, yeah, there's even partitioning and uneven partitioning at these larger volumes. So that's what happens at the macro scale. So then the question is, can we make these types of multi-material 3D structured particles, and many of them at the microscale? So that's a challenge that that we've been addressing using a technology that we call transient liquid molding. And so this combines two technologies. One is inertial flow sculpting, and the second is an automated photopolymerization system. So in inertial flow sculpting, I'll tell more about this in following slides. We make use of the fact that around pillars, we can modulate the shape of a flow stream. And so we could co-flow two polymer precursors, and then structure those polymer precursors into a shape like this C shape. And then we can design a mask, and then we can expose those polymer precursors to UV light, for example, to photo cross-link them and create this structured particle. Okay, so I'm going to talk a little bit about this inertial flow sculpting, because we developed some software in order to know what types of microfluidic devices we would need to construct in order to create this flow sculpted shape of interest. Okay. So one of the challenges with this is how do I know what sequence of pillars are going to create the deformation that leads to the flow shape that I'm interested in creating? And so we used the genetic algorithm, an approach to do optimization that's based off of developing a chromosome of configurations, where each chromosome has the inlet flow conditions and the pillar sequence, then leads to some phenotype, which is the outlet flow configuration. And so here's an example. You have an inlet flow, which shows the blue region. And this is a cross-section of the flow where one particular material is. And then you have a particular sequence of pillars that is used to deform that shape. And then you have the phenotype, or the outlet flow, what that shape looks like. And so we can input into this type of algorithm different types of mutants of this pillar sequence and inlet flow, and then we see what the outlet flow conditions are. And then we have a fitness function that says, okay, how close is this to some particular flow shape that I'm trying to target? Let's say this U shape. And then, that fitness function would say, the top two of those shapes, the diamond-like shape and the H-like shape, are not as close to the U shape as the bottom shape. And so that fitness function would then say, okay, the bottom shape is one where we should inherit more of that. We should have that chromosome stay around, mutagenize it a bit, and then keep iterating until it gets closer and closer to the shape of interest. And so this is how we optimize and solve this inverse problem of, okay, if I have a shape of interest in the flow, how do I generate the pillar sequence that will lead to the flow deforming to create that shape of interest? And so we have developed a software called FlowSculpt. This is work by a postdoc in the lab, Dan Stokeline. And you can download FlowSculpt at this website here. And basically it gives some ability to do drawing, to draw the flow shape of your interest. It has then output capabilities to output a CAD design of the microfluidic device that would create that particular flow shape of interest. So how does this work? So an example that we've prepared is can FlowSculpt generate these letters for UCLA? So these are the target flow shapes, and can FlowSculpt create pillar structures that lead to these target flow shapes? And so the results that you get from the simulation are pretty good, right? So this is the U. You see that matches pretty well with the target shape. You see that here is a C, here is an L, and here's an A. So, it is not exactly the letters, maybe more like the script of some serial killer or something like that, but still a pretty good approximation of our target shapes, given that we're manipulating the flow just with very blunt operators of pillars in a channel. And so you can really get these types of shapes, and that occurs in ways that are really not intuitive if you were trying to design where these pillars were in a forward type of design process. So making use of the genetic algorithm is very critical in getting this to work in a functional way. So does this match with what we see experimentally? The answer is pretty well. So this is looking at confocal imaging of fluorescent dye that is flown into a channel. So we're looking at the channel cross sections, and you see that the channel cross sections match pretty well with the predicted results for these particular letters. And beyond single material, so you saw this blue material in a white background of other flow materials, we can also predict the flow shapes for and design in a design pillar combinations that lead to flow shapes of multiple materials. So in this case, you see two materials, and our output is a diamond-shaped pattern that has one half of the blue material and one half of the red material. And so here's some again using FlowSculpt to solve for desired flow shapes. And you can see the results on the right. So the left is the target flow shapes of 2 to 3 different materials, red, blue, and green materials. And you see the FlowSculpt results on the right, demonstrating the ability to create geometries that are similar to those materials. Okay, so once we had this capability to design these types of U-shaped particles that have an internal region containing a hydrophilic material and the external region having a hydrophobic material, we were able to then manufacture these types of particles in a microfluidic device. And so, of course, we want to manufacture them at small scale and at high throughput. And so this is a particular design for manufacturing polyethylene glycol and polypropylene glycol materials to create these amphiphilic particles that can hold droplets. And we use parallel masks in order to polymerize an array of these particles all at one time. And then we repeat the process of flowing to create the shape, stopping the flow, and then cross-linking through the mask in order to generate a number of particles. And so here's an example of the particles that we fabricated. You can see there's pretty high uniformity in the shape of the particles. You can kind of see an internal region that's the polyethylene glycol that has slightly different contrast than the external region, which is polypropylene glycol. And you also see the length scale here. So the scale bar is 500 microns. And so the cavity here is in the 100 micron scale, and the total particles is in the few hundred micron scale. Just if you zoom in a bit more, you can see both the outer polypropylene glycol layer and the internal PEG layer. The PEG layer is here dyed with a fluorescent dye. And so you can see that the internal layer lining the particle. You can also, what we're showing on the bottom is a particle of a certain dimension, but by changing your mask, you could also change the dimensions of the particles that you polymerize. So you have capability to create three-dimensional shaped particles that have both the shape of the flow stream as well as the shape of the mask that you apply with light that filters the light. And so what you can create then is an extrusion of the mask shape with the flow stream shape, and that creates your final particle shape. Okay, so once we have these particles, can we then make these uniform drops just with a simple shaking procedure, right? So that's that goal here. We don't want any microfluidics for the end user, right? Microfluidics can be used to make these particles, but the end user now should have the ability to take the particles that are provided to them with their sample of interest, shake it up or pipette it up and down using kind of standard instruments in any lab, and create the monodispersed compartments that are the key to driving a lot of these quantum diagnostic assays. And so what you see here is kind of our assay. You have water, these particles, we call them drop carrier particles, and oil that are in a vial. We then do a simple mixing and centrifugation process, and that creates what we call particle drops, which are these particles that are supporting the water phase internal. And this works, and we see that we can create uniform drops. And you notice that these drops are not spherical, right? So they are supported by the particle, and so their lowest energy state is not a sphere like a normal drop in an emulsion, but this kind of cat-eye-like shape that is because they're templated by these particle carriers. Okay, so we started to study these particle drops a bit more. And so one kind of important point is are they monodispersed? Do they have very uniform volumes? And so what we start to see is in different oils. So you have in the blue and yellow, two different oils that we're using to create these particle drops. The aqueous solution is the same. And you see that there's a mode in both of those oils that is really uniform compared to a droplet that is just stabilized by surfactant that spreads out over orders of magnitude larger diameters. And so that's because the surfactant, although it stabilizes a droplet to coalescence very rapidly, it doesn't create a preferred volume that the drop carrier particles do create. As I mentioned, that these drops are not spherical, and so one key aspect is their shape and how uniform their shape is, and you can kind of quantify these things, and you see that their shapes are also have circularities that are not like a sphere, not one which you see with surfactant stabilized drops, but are closer to 0.5, or depends. Obviously, depends on the shape of the particle. And the filling of these particle drops, as we saw in the theory that suggests that you would create a mode in volume, depends on the volume that you apply to the droplets. But after a critical volume, what you start to see is that they all start to fill with this mode about 200 microns for these particular drops that we generated. And these types of drops that are formed are emulsions that have particles surrounding a droplet, which we're calling an armored emulsion, appear stable over many days. So this is over 3 days, and you can see that the nominal diameter, the mode and the diameter stays very consistent over those 3 days. You can see the distribution is pretty consistent over those 3 days for these particular drops. Here are some pictures that kind of showing the stability of the drops at day 1 and day 4. We're showing a fluorescent dye that's staining the volume of fluid in the droplet so you can see them. There's some unique features of these types of systems, in that you can prevent the crosstalk between droplets because they're surrounded by this solid phase. So here's an example where we had 2 different solutions that were created. And one had a red dye and particles supporting droplets with the red dye, and the other solution had a green dye, and we created these particle drops that supported a green dye, and then we mixed those 2 together. We agitated them, and what we see is that there's no crosstalk. We don't see particles then that have, at least with that have red and green, and we don't see loss of green or red in droplets. So there's stability, but also you don't have drops coming off and out and in, and you don't have the droplets cross-talking across separate particle drops. So that's an interesting aspect that may be different than the surfactant stabilized systems, where you may see some crosstalk that's facilitated by the surfactant itself. So what can we start to do with these particle drops? Well, anything that you can do with a microfluidic drop, basically, or anything you can do with a microwell array. And so here we're showing kind of 2 examples, proof of concepts. On the left, we're showing the filling of these particle drops with particles. So in this case, we chose different size particles because we wanted to show that there's capability to fill, but also prevent the filling or the encapsulation of a particle if its size is too big. So in this case, we could encapsulate particles of different sizes into the particle drops, but if the particle size is larger than the volume that's held by that drop, then it wouldn't be encapsulated. And on the right, we're seeing a time-lapse image of an algae that's encapsulated in another particle drop, showing the viability of that cell over days as it's growing and moving in the drop. We can also perform reactions in these drops. So here's an example of an immunoassay type reaction where we're amplifying the presence of a protein, and that amplified presence is read out using a fluorescent marker of the amplification reaction. And one interesting thing we see here also is that amplified fluorescent marker accumulates in the particle, the PEG region of the particle, at a higher concentration than the aqueous portion, and so that could potentially even amplify the signal even further. So I think there's a potential here also to leverage the reactions and what can occur on the solid phase and aqueous phase to improve immunoassays in these types of systems. Okay, so understanding that these types of new particle drop systems had these unique capabilities, we also looked into literature and found, with any good idea, there's some folks that have explored some aspects of the idea previously And so this is work from Richard Matthey's lab, where here they're using a microfluidic device to encapsulate cells in agarose particles, agarose drops that are then solidified. And they use those particles as a way to template drop formation. Okay. Of course, here there's still microfluidics that are needed in order to put the cell into the droplet. And so that's the part that is still an issue with this approach. But we wanted to build off of the ability of that they demonstrated for here a spherical agarose particle to template emulsion just with simple mixing and shaking. And so we've developed a way that we could get rid of the microfluidics for this type of process. And so we could template monodisperse emulsions by mixing and shaking spherical particles that had cavities embedded in them. So those cavities then could hold cells, where the previous approaches you needed microfluidics in order to put the cells into the particles. And so you can see here, this is not a microfluidic generated emulsion. This is an emulsion that's generated using the particle and just mixing with simple processes. And you can see that because there's some of the droplets that also have more than one particle. So you can see a triangular shaped droplet has 3 particles and in a few droplets that have 2 particles together. But generally this is, at least a very monodispersed drop sizes. Here's a fluorescent image of this type of emulsion where you see the red dye, red fluorescent dye is staining the particle that has this cavity that holds cells. And the blue fluorescent signal is showing the aqueous phase that's surrounding the droplet. And you can see that there's pretty good monodispersity there, and I'll show quantification of that in a later slide. So the idea here is this, what we're calling a dropicle, can be used, for example, in single cell analysis workflows. So in this case, we're using this dropicle to then put it into a well plate, seed cells, attach the cells to the dropicle. We can then use a simple process of emulsification using pipetting to encapsulate the drops. And then we can read out some signal, capture that signal on the particle, and then read it out using standard flow cytometers. And so this is really the workflow. And again, there's no microfluidics. There's all processes that are standard to research labs and clinical labs. And that's really the goal here. So how do we manufacture these types of hollow particles? So we use a aqueous two-phase system and microfluidics itself, again, to do the manufacturing. So here we have a PEG dextran system that then phase separates. The PEG is a photocross-linkable PEG, and that is after the phase separation, we can cross-link that. We can then wash out the dextran as we transfer it back to water, and that gives us these shell-shaped particles. So when we then create emulsions with these particles, they are pretty monodisperse. And so you can see the emulsion picture of it on the right side. On the left side, you can see that we have particle drops that are formed, and you can see that there are 2 regions. So one are the drops that are templated by the particles, and they have very uniform size, CVs around 5%, which is it's quite good and comparable to many microfluidic approaches to generate droplets. And then you have satellite droplets that have no particles inside, and those are can be avoided because they are not. You don't have a solid phase to capture signal on them, and so they don't will not show up in the final assays with flow cytometers. So cells can be loaded into these systems. So you're seeing a live cell stain on and particles that are in red and cells in green, and you can see that they remain viable over a day period at least in these emulsion systems, particle-supported emulsions. And you can load cells just as you would in other droplet systems following Poisson statistics, depending on really the concentration that you apply to begin with. We can then run assays on cells that are encapsulated in the droplets, just as you would in a microfluidic or another type of system that has a solid phase. And here we're capturing the secretion from a cell in a droplet onto the surface of the particle that's creating the droplet. And then after we then bring that back into an aqueous phase, we can then stain for the presence of that secretion, and while maintaining the cell on that particle, and then we can sort based off of both the secretion and aspects of the cell using standard flow cytometry systems. So this is an exciting kind of demonstration of how this type of technology can be used in single cell analysis. Obviously, we're at kind of the initial stages of using these types of technologies, but the capabilities are there. Really, it was about, can we create uniform droplets with simple processes? And we've also shown that we can analyze these types of particles with flow cytometers. So here's the question really the direction that we're going. We're going from this idea of lab on a chip, which there is a chip, but around that chip there's a lot of infrastructure and instruments. And so that leads to a barrier in adopting new technologies and trying out new technologies in the clinical labs as well as in research settings. And we're going from that lab on a chip to lab on a particle. And that's really driven by this economic approach to make monodispersed droplets just by simple shaking operations. Then these types of droplets, the signals from the droplets can be captured on particles that can be read with standard flow cytometers. And so you avoid the need for new instrumentation, new infrastructure on that side of things as well. And that solid phase, I think, is particularly interesting because now you can do new engineering of that solid phase, and that will open up new opportunities for what I'm calling lab on a particle systems. And I think these lab on a particle systems can help accelerate our understanding and our of life science. So as a life science research tool, for example, discovery of biological rare elements as we look at single nucleic acids and single proteins that might be present in different disease states, as a way to do single cell analysis at a much higher rates and at lower costs. So for these human cell atlas type applications, but across also a number of applications in health and energy and environment, in creating new approaches to evolve cells or other products based on sorting, based on secretions or other aspects that can accumulate and be measured in these types of small compartments. So I think that there's going to be a lot of new technology developments that are needed as we advance these lab on a particle systems. And one of the key metrics that we'll start to, I think, pay attention to is really the number of single cells or molecules that we can analyze in an experiment. And so maybe this is our next metric, similar to Moore's law on transistors, that we should be paying attention to. And I think that the technology will accelerate rapidly using these types of lab on a particle systems so that we can get orders of magnitude higher numbers of single cells and single molecules, really this quantum level of information that is the key to understanding biology and understanding disease processes. So with that, I'm happy to acknowledge really the contributors from my lab and as well as our collaborators who have laid the foundations for this new approach, which I'm extremely excited about. And I thank you, the audience, for listening, and we'll be happy to answer questions through the online process. Thank you very much.
Thank you, Dr. Di Carlo, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. As a final reminder, our speaker will follow up with any questions you've submitted via email. That is all for now. Thank you for joining us. We'll see you next time. Goodbye.